My lab is part of CellSite. CellSite is the Ocular Stem Cell and Regeneration Program in the Department of Ophthalmology. And we use human-induced pluripotent stem cells to generate these mini human retinas in vitro, which we call retinal organoids. And these mini retinas recapitulate the whole histoarchitecture and composition of a normal human retina. And so they have a lot of potential. They can be used to model diseases, and those models can be used to screen for drugs and to test their efficacy in a human central nervous system model, right? But for that to be true, the retinal organoids need to really recapitulate at least some of the features, the pathophysiological features that you would see in that disease. And that was not available for retina so far. So what we did was we took human iPS cells derived from patients that had familial forms of Alzheimer's disease. And we used those iPS cells to generate these mini human retinas in vitro. And we found that these retinas have the whole cellular composition and structure of a normal retina, but we found as early as 100 days of differentiation, which for a human retina in vitro is not very long, we found a very severe increase in deposition of amyloid. That seems to be amyloid plaques because they co-stain for amyloid beta and NIAD4, which is a staining for amyloid. And we also saw an increase in tau phosphorylation in this model. So using this model, we also developed a paradigm for drug screening by coupling this disease model with a technique to quantify amyloid plaques in whole retinal organoids that can be used in longitudinal studies to track over time what's happening in these organoids. And so we're planning on applying that to the testing of potential drug candidates.